When you take a pill for a headache or a daily medication for a chronic condition, you trust it will work. You believe it is safe. This trust is not accidental. It is built on a foundation of rigorous science and careful testing. Every single medicine, from a simple painkiller to a complex cancer treatment, undergoes a huge number of checks before it ever reaches your pharmacy. This process is essential to modern healthcare. It ensures that the medicine you rely on contains exactly what it is supposed to, in the exact amount needed to help you, without harmful impurities that could make you sick. The goal is always to protect your health and well-being. Behind the scenes, in laboratories all over the world, scientists work tirelessly to guarantee this safety and effectiveness. They are the unseen guardians of your health. Their job is to develop and confirm the testing methods used to check these drugs. Think of it like a chef perfecting a recipe. Before they serve a dish to hundreds of people, they must be sure the recipe works every single time, no matter who is cooking. In the same way, pharmaceutical companies must be absolutely certain that their methods for testing drugs are reliable and give the correct results every single time. This is not just good practice. It is a fundamental requirement for making safe medicine. This commitment to testing is what separates modern medicine from the remedies of the past. It provides a shield of protection for every patient. Imagine if a tablet that was supposed to contain 500 milligrams of an active ingredient only had 300 milligrams. It might not be strong enough to treat your illness, leaving you to suffer longer. Or worse, what if it contained more than the intended dose? This could lead to dangerous side effects. This is why the testing process is so critical. It acts as a quality control system, catching potential problems long before a medicine is packaged and sent to the people who need it most. The journey of a medicine from a laboratory concept to your medicine cabinet is long and complicated. It involves discovery, development, clinical trials, and finally, manufacturing. At every single stage of this journey, testing is a constant companion. The methods used for this testing must be proven to be dependable. They must be validated. This validation is a promise from the manufacturer to you, the patient, and to the regulatory bodies like the FDA. It is a promise that every batch of medicine meets the highest standards of quality, safety, and efficacy. It is the science that allows all of us to have confidence in the treatments that keep us healthy. So, what exactly is this critical process? In simple terms, analytical method validation is the process of proving that a specific testing procedure is suitable for its intended purpose. It is like creating and testing a detailed rulebook for a scientific measurement. Before a company can use a method to test a drug, it must first demonstrate that the method is reliable, accurate, and consistent. This involves a series of carefully designed experiments that produce documented evidence. This evidence shows that the method will always give trustworthy results when used correctly. It is the scientific proof that stands behind the quality of every medicine. Think about a simple example, like testing a tablet for a common pain reliever. The company needs to know exactly how much of the active ingredient, such as paracetamol, is inside each tablet. They develop a specific laboratory procedure or method to measure this. Method validation is the process where they prove this procedure works perfectly. They confirm that it can accurately measure the amount of paracetamol without being confused by other ingredients in the tablet, like the binders and coatings that hold it together. This validation process ensures the test is not just a guess, but a precise and verifiable scientific measurement. This rulebook is essential for consistency, especially in a global industry. A medicine might be developed in one country, manufactured in another, and sold in dozens more. The testing method must produce the same, correct result, no matter which laboratory is performing the test. Method validation creates a universal standard that everyone can follow. It ensures that a tablet tested in a lab in India is shown to have the same quality as one tested in a lab in the United States. This global consistency is fundamental to public health, ensuring that patients everywhere receive the same high-quality medicine they expect and deserve. Ultimately, method validation is about building confidence. It gives the pharmaceutical company confidence that its manufacturing process is under control. It gives regulatory agencies like the Food and Drug Administration confidence that the company is meeting all required quality standards. Most importantly, it gives doctors and patients confidence that the medicine is safe and will perform as expected. Without this formal validation process, Drug testing would be unreliable. It would be impossible to guarantee that each pill, capsule, or injection meets the strict specifications necessary to protect patient health, making it a cornerstone of modern pharmaceutical science. The process of method validation is built on several key characteristics, which I like to call the 5 pillars of trust. The first of these is specificity. This means the test method must be able to detect and measure only the specific substance it is looking for, which is usually the active pharmaceutical ingredient. It must not be fooled by the presence of other chemicals, such as impurities, byproducts, or the inactive ingredients in the medicine. For instance, when testing a headache tablet, the method must specifically measure the painkiller and ignore the binders, fillers, and coating materials that make up the rest of the pill. This ensures the result is pure and not distorted. The second and third pillars are accuracy and precision, which are related but distinct. Accuracy refers to how close the test result is to the true, actual value. If a tablet truly contains 500 milligrams of the active ingredient, an accurate method will give a result very close to 500 milligrams. Precision, on the other hand, refers to how consistent the results are when the test is repeated multiple times. A precise method will give nearly the same result, say 499 milligrams, 501 milligrams, and 500 milligrams, over and over again. A good method must be both. It must be consistently correct, not just consistently wrong. The fourth pillar is linearity. This principle ensures that the test results are directly proportional to the concentration of the substance being measured. In simpler terms, if you double the amount of the drug in a sample, the test result should also double. Scientists check this by preparing samples with a range of different concentrations, from low to high, and plotting the results on a graph. The outcome should be a straight line, which proves the method is reliable across the entire expected range of dosages. This is crucial for testing different strengths of the same medicine, like a 10 milligram tablet versus a 20 milligram tablet. Finally, we have the fifth pillar, robustness. A method is considered robust if it continues to provide accurate and precise results, even when there are small, deliberate changes in the test conditions. For example, what happens if the laboratory temperature changes slightly, or if a different batch of a chemical is used in the test? A robust method will not be significantly affected by these minor variations, which are common in real-world lab environments. This ensures that the test is dependable and can be successfully transferred and used by different analysts in different labs without failing, making it a practical tool for global quality control. The rigorous process of method validation provides direct and powerful benefits for the most important person in the healthcare chain, the patient. When a testing method is properly validated, you can be confident that the medicine you are taking has the correct dose. This is critical for its effectiveness. Too little of an active ingredient can mean your condition goes untreated, while too much could cause harmful side effects or even be toxic. Reliable testing also screens for dangerous impurities that might have been created during the manufacturing process, ensuring the product is not only effective, but also pure and safe for consumption. This assurance protects everyone, from a child taking an antibiotic to an elderly person managing heart disease. For example, for drugs that have a narrow therapeutic window, where the difference between a helpful dose and a harmful dose is very small, accuracy is a matter of life and death. Medications for epilepsy or blood thinners are prime examples. Validated methods ensure that each batch of these critical medicines is exactly within the safe and effective range. This meticulous attention to detail is what allows patients and their doctors to trust that the prescribed treatment will help, not harm, building a foundation of safety in medicine. While the primary benefit is patient safety, pharmaceutical companies also gain significantly from investing in robust method validation. A well-validated method is efficient and reliable, which helps to prevent costly manufacturing errors. If a batch of medicine fails a quality test due to a faulty method, the entire batch may have to be discarded, leading to millions of dollars in losses and potential drug shortages. By using proven, robust methods, companies can avoid these failures, streamline their production, and ensure a consistent supply of high-quality medicine to the market. It is a smart business practice that aligns perfectly with public health goals. In conclusion, Analytical method validation is not just a regulatory hurdle, it is a fundamental scientific discipline that upholds the integrity of the entire pharmaceutical industry. It is the bedrock upon which drug safety and efficacy are built. The continuous development and application of these strong testing methods are essential for protecting public health. As science evolves, so too will these methods, always aiming for greater accuracy and reliability. This ongoing commitment ensures that every patient can trust the medicine they take, a shared benefit that strengthens both the health of our communities and the companies dedicated to serving them.